Hi, this is B. Charanjivara, Physics Lecturer APRJC Thatpudi. In this video lesson, I will teach moving charges and magnetism. See, in this particular chapter, in 1820, Oersted discovered and observed the relation between moving charge and magnetic field. So, according to his observation, if current is flowing through the conductor, the magnetic field produced around the current conductor. This is the observation made by Oersted. That is why it is called Oersted law. So what is Oersted law? An electric current creates magnetic field around itself. Suppose if this is, you consider that this is a current carrying conductor, and if current I is passing through the conductor, then a magnetic field will produce around the current carrying conductor. This magnetic field are circular in shape. They are closed circular loops, concentric circles. They are concentric circles, and the center is at the position of the conductor. And the magnetic field and the current, they are perpendicular to each other. And this is the circular plane, and the current acts the normal to the plane of the magnetic field. So the direction of the magnetic field gives the right hand thumb rule. So this is the right hand thumb rule. See, you observe, this is the right hand thumb rule. Then the conductor is holding with your right hand. The direction of the current indicates the direction of thumb along the direction of thumb, and the rest of the fingers folded in a particular direction gives the magnetic field. So if the current is moving in this way, upward direction, then the direction of the field is representing the direction of the rest of the fingers folded. So if suppose this is the way the direction of current is moving down, and the rest of fingers folded in the other direction. So this is how we can observe the direction of magnetic field using right hand thumb rule. This is right hand thumb rule. So this is the direction of the magnetic field. This is the direction of the magnetic field in this case. And suppose if the current is reversed in this direction, then the magnetic field, see, just imagine this is the direction. Then what happened? The direction is like this. So magnetic field direction. So magnetic field direction depend on the direction of current. So a current carrying conductor produces magnetic field. A current carrying conductor produces magnetic field. Magnetic field around itself. And the direction of the field can be identified with the right hand thumb rule. Right hand thumb rule. Right hand thumb rule. Or we can also, this can be identified as screw rule. Screw rule. Suppose if you imagine that this is a screw, if this screw is trying to rotate in this direction, it moves up. If this is the direction, anti-clockwise direction, it moves up. If clockwise direction, it comes down. So you can also explain this with corkscrew rule. That is what is called screw rule. And this is all regarding Oersted's law. And now let us observe the another important. See, the next one is magnetic field. The magnetic field. So what is actually a magnetic field? It is the space around which it has its influence. That is what is called field. Now let us see. It is the space around the magnet or current carrying conductor around which magnetic effects can be experienced. This is what is called magnetic field, like gravitational field, electric field. And we can represent this field with magnetic intensity of magnetic field. Intensity of magnetic field. This can be measured. Intensity of magnetic field. So this intensity of magnetic field represented with V, it is a vector. This is a vector. It is a vector quantity. So actually magnetic field intensity B is force acting on unit north pole. Force acting on unit north pole. Force acting on unit north pole. At that point in the field. At that point in the field. In the field. So, the magnetic induction is a vector, since force is a vector. So, B acts as B, force is a vector. And now, force experienced can be written as mB. So, what is the unit of B? B, unit of B, Newton per ampere meter, one unit. And another unit is Tesla. One more unit is Weber per meter square. These are all the units of magnetic intensity or magnetic induction. It has a CGS unit. The CGS unit of B, magnetic field intensity, is Gauss. It is the CGS unit of magnetic field, Gauss. And next, one Gauss is equal to 10 power of minus 4 Tesla, the conversion. These are all SI units of B. SI units of B, magnetic field intensity. This is all regarding magnetic field. And now let us observe another concept. Say moving charge and magnetic field. A charge can produce magnetic field if it is in motion. So, if a charge is moving, it produces magnetic field, like a spinning electron. A spinning electron produces magnetic field, a dipole. A dipole due to spinning electron. Similarly, if a moving charge is entered into the magnetic field, it also interacts with the moving charge. That is why there are two concepts under consideration. A charge can produce magnetic field if it is in motion. For example, if the electron, a spinning electron produce dipole, say north pole and south pole, the spinning electron. And if the charge is moving in a magnetic field, it interacts with the moving charge. Suppose if a charge is moving, say electron, you can consider electron or proton. If it is moving, a positive charge is moving with certain velocity. Moving means it is having certain velocity v, not at rest. When it is moving in the magnetic field, say this is already field due to the magnet, then what happens? It deflects in a circular path. So, this magnetic field can also interact with a moving charge. So, these are the two concepts regarding moving charges and magnetic fields. Now, let us see the magnetic force on moving charge. So, if a charge is moving in a magnetic field, it undergoes deflection or it is being influenced by the force. So, the force experienced by the moving charge due to magnetic field is equal to Q into V cross B. This is the cross product of the two vectors, velocity of the charged particle and the magnetic field. Suppose, let us consider a charge is moving in a magnetic field. I just write down, this is the direction of the magnetic field. And if the charge is moving in this way, this is the direction of the moving charge with certain velocity v, which is making angle theta, with the direction of the magnetic field. Then what happened? The force. So you can represent the force on this charge as a cross product of v and v. Suppose you represent that this is the magnetic field intensity and this gives the velocity of the charged particle. If these two are separated by an angle theta, then the force experienced is given in the direction normal to the plane of the two vectors B and V. So this gives the force. Force is equal to Q into this V cross B. I just write down V cross B. V cross B. And if you consider that there is a unit vector in the direction of force, then the force experienced is equal to magnetic force on moving charge F equal to QVB sin theta. This is the magnitude of force, and if you consider this as a vector, and this force acts in the normal direction with a magnitude of QVB sin theta. So this can also be expressed in terms of the thumb rule, right hand thumb rule. This can also be expressed with the help of right hand thumb rule. And just you represent the thumb, index finger and middle finger mutually perpendicular, or you can also make some angle between the two vectors, any two fingers. See, one is normal to any two of the fingers. So if the direction changes, automatically the direction of force will also change. So we can explain this also with the help of this right hand thumb rule. Yes, this is a cross product. We can explain this with the right hand thumb rule. So force is equal to, the magnitude of the force is equal to QVB sin theta. So if, now let us see, if theta is 90 degrees. If theta is 90 degrees means this is the magnetic field. And the charge is moving normal to the direction of the magnetic field. So the angle between the B and V is 90 degrees. Under this condition, force experienced by the charge, moving charge, is equal to, this is maximum. Since theta is 90, this becomes maximum. Its value is QVB. QVB. And if theta is 0, that means 0 degrees. That means the magnetic field B and the charge is also moving in the same direction. Then what happened? Theta is 0. So there is no effect of force on the moving charge. So this is, say, minimum is equal to 0. Sin 0 is 0. There is no effect when the moving charge and the field are parallel to each other. And if both are perpendicular to each other, then the force is maximum. So if both are making some angle theta, then force will be QVB sin theta. So these are the different cases that magnetic force on the moving charge. Now let us see, when V is perpendicular to B, suppose you imagine that the magnetic field is into the board. So into the board we can represent with the cross. So magnetic field lines are into the board, going into the, towards the board, can be represented with cross. And if the field is coming out of the board, then we can represent with dots. So there are two notation. If the field is going into the board or paper, it is represented with the cross, and if it is coming out of the board, it is represented with some dot. So this is the notation of the magnetic field. If a dot means it is coming out like this, it is coming out. And a cross means it is going into the blackboard or whiteboard. So when the field is in this direction, if the charge is moving somewhere here, normal to the direction, then what happens? If the charge entered to the field with certain velocity, then it starts with somewhere here, see at a point P, and it is moves in a circular path. It moves in a circular path. It moves in a circular path of radius r. It moves in a circular path of radius r. So at any instant, see, this is the direction of velocity at this particular position. And the force will experience towards the center of the circle. So this is how the velocity here and the force will act in this direction. Now, what is the radius with which it deflects in the perpendicular field? So we know the centrifugal force is equal to mv square by r. So the radius can be written as mv square by f. But f is equal to q into v into b. Suppose you substitute here, r is equal to mv square by qvb. So v square cancelled. So the radius is equal to mv by qb. So this is the radius of the circular path through which the charge moves in a circular path. So r depends on the velocity of the charge and the magnetic field intensity, that radius. More the velocity, more will be the radius. Isn't it? So this is how we can find the radius of the circle when V is perpendicular to B. If V is moving, making some angle with theta, making some angle theta with magnetic induction other than 0 degree centigrade. Let us see what happened. Suppose if the moving charge V is making an angle with B other than 0 degree centigrade. 0 degrees are normal. Suppose you consider this is the direction of the magnetic field and this is the y-axis and this is x-axis. Now the charge is not moving along B or perpendicular to B, but it is moving in with certain direction which is making angle theta with certain velocity v. Then this velocity of the charge resolved into two components. One is the velocity along parallel direction along x-axis, and this is the vertical component, the perpendicular component. So the two components are parallel component and perpendicular component. And now we know that if theta is 0, regarding this component, parallel component, which is along the direction of B, has no influence. It has no influence. It moves as it is. But whereas, if you consider this particular component of velocity of the charge, it is normal to the direction of the magnetic field. That is why this particular part execute in a circular path, like this. So it keep on moving like helical path. And in one particular time, in one cycle, this charge moves from this point to this point. This is what is called pitch. This represents pitch. So in one time, in one particular complete cycle, it moves from this point to this point. This is called pitch. This is the horizontal distance traversed by this vertical component of the velocity of the charge. Whereas the horizontal component continues to move in the same direction as it has no influence, and it's become force on this is 0, and the only thing is it's moving. Now the path of this vertical component is helical. Then what is the radius of the helix? R is equal to, as you know the formula, R is equal to, and the pitch first you write down, pitch equal to actually horizontal distance. Pitch is equal to the velocity in the direction of the magnetic field multiplied by time. So this is equal to 2 pi m into velocity in the parallel direction by qb. So this is the pitch of the helix, the charge which is moving along horizontal direction. And now let us see another important concept. Now let us see what happens if a charge is moving both electric and magnetic fields, the combined fields. Now let us see the difference between magnetic field and electric field. Say for example, as far as electric field is concerned, this is electric field, and I will write here it is magnetic field. So in electric field, we know the electric field intensity is equal to force acting per unit positive charge. But in case of magnetic field intensity, it is the force acting on unit north pole. So in this case, force is equal to EQ. But in this case, force is equal to Q into V cross B, or this can be written as QVB sin theta. You consider only its magnitude. Now what we can infer from this formula is, see the force experienced or in the electric field, it does not depend on the angle or whether it is rest or in motion. So the charge may be whether it is at rest or in motion. It has influence of force. Whereas in case of magnetic field, force due to magnetic field, it is influenced only when the charge is in motion, since it has to have some value of velocity. And it also depends on the angle. So these are the main differences between electric field and magnetic field. And in electric field, and a isolated charge can exist in electricity, whereas in magnetism, isolated pole is not possible to exist. So always magnetic poles exist in pairs, north and south. Now this is the major difference between the electric field and magnetic field. Now suppose if a charge is moving, with certain velocity v in mutually perpendicular electric and magnetic fields, mutually perpendicular electric and magnetic fields. I write here, this is the magnetic field. So I write this is magnetic field intensity. So if these two are perpendicular, now let us see the direction of force. It is influenced by force and it moves in this direction. So in this direction, it moves in this direction with certain radius r. So what is the force due to magnetic field? alone, magnetic field alone, is equal to QVB. Since theta is 90, that is the force due to magnetic field alone. And suppose if you apply the normal electric field also in this direction, the normal electric field, I write here, this is the normal direction electric field also applied, plus and minus. So I write here somewhere here like this. So I apply the normal electric field also. So this is the electric field lines with E, electric field with E. Then what happened? Force due to electric field alone is equal to Q into E. So if we represent this in a three-dimensional coordinate system, we can just represent this in a three-dimensional coordinate system as, say, this is the X axis, and this is Y axis, and this is Z axis. And now you represent the velocities along X axis, and magnetic field is along Z axis, and electric field along Y axis. So how do you write now this one? See, E bar can be written as E bar is equal to, just write down, E into, in which direction this is? A unit vector in Y axis. And now velocity component. Next one is B. B in which direction? B in which direction? B multiplied by Z axis. So I write here K cap. And another one V. V is equal to, in which direction? V into I cap. So these are the three different vectors acting in three mutually perpendicular directions. That is I, J, K. Now force due to this one, electric field, Fe is equal to QE, So this you can write down as Q into EJ. This you can write down as Q into E into J cap. This is the force in the direction of Y axis. So this is the electric field in this direction. And now what will be the force due to magnetic field? You can write down force due to magnetic field is equal to Q into V cross B. V cross B means V Q into V into I cap cross B into K cap. So this is the force due to magnetic field. Now what happened? You can represent this as force due to, this will be acting in this direction, magnetic field force. Force due to magnetic field will be acting in this direction. This is the cross product of V and B, acting in the negative Y axis. That is why the force due to magnetic field can be written as minus QVB into, what is the direction? In this way. So I write here minus J cap. So these are the two forces. First one is force due to electric field and force due to magnetic field. Then what is the net force here due to these two? So the net force is E in this direction minus this one will give you the net force. So net force is equal to, now the net force is equal to force due to electric field plus force due to magnetic field. So I write here Q into force due to electric field is how much? Q into E minus VB into J cap. This is the net force acting on the moving charge when it is moving through mutually perpendicular electric field and magnetic field. So this is how we can represent the net force. So force acting this due to electric field and this is due to magnetic field, and the difference of these two give the net field E minus VB J cap. See, this is how we can get the, and now this is what is called Lorentz force. This force is called Lorentz force. This force is called Lorentz force. And we can also write down the Lorentz force as, now the Lorentz force can be written as this one. Lorentz force is equal to Q into E plus V cross B. This is what is called the Lorentz force, Lorentz equation. Now, if we make the two fields in such a way, we apply the two fields, electric field and magnetic field, in such a way, the charge undeflected. That means force due to magnetic field and force due to electric field, we have to make same. So if you just apply electric field alone, it bends in downward direction. And if you apply magnetic field alone, it bends in the upward direction. And if you simultaneously apply the two forces, it neither bend upward nor bend downward, and it goes as it is. So under this circumstance, let us see what happened. Now what we are doing is, a charge is moving with certain velocity V in the electric field. So I write here, because of this electric field, it bends in this direction, because the positive charge attracted towards the negative charge. So this is how it bends in this direction. So this is the electric field. So it bends with certain force. The force here given as, force due to electric field is given as E QE. And I simultaneously applied magnetic field. Magnetic field normal to this direction of the charge. So I just applied another force, magnetic field I applied here. And then what happened? This bends in the upward direction. So What is the force due to magnetic field? QVB. And I made the two charge fields in such a way, this is undeflected and this is going straight. That means, what is the condition? Under these circumstances, I just made Fe is equal to Fm. Therefore, I just write down Q can be is equal to force due to electric field is equal to force due to magnetic field, QVB. Then what happened? Q can be cancelled. So, velocity is equal to E by B. So now, the crossed electric and magnetic field will serve as velocity selector. See, velocity depends on electric field and magnetic field. Now, this V, this crossed electric and magnetic field will serve as velocity selector. So, I write here, crossed electric field E and magnetic field B will serve as velocity selector. This is a very important concept. The crossed electric field E and B serve as velocity selector. This can be obtained using Lorentz formula. So we can find the magnetic intensity of the magnetic field due to a current carrying conductor at any point P, at a distance P in a particular medium with the help of Biot-Savart law. So, according to Biot-Savart law, yes, we can determine the magnetic field intensity due to a current carrying conductor. So, for this, what we have to do is, just you imagine a wire, thin wire, curved wire, and you consider a small element here. Just you consider a small element here. Small element dl, current. And this element is, see, a charge has to flow, it has to move with certain velocity, it has to flow. So it has some length of some part, it has to travel some distance. That is why we have to consider a small element dl. Such dls are combined to form a long wire. So in this wire, the current is flowing, and the direction of the current, along the direction of the current, you take the direction of the element dl. That is why we call it as dl bar. And you consider any points, see somewhere here P. I just consider some point P here in the medium. And this particular point is making some angle theta. I write here the position of this point is represented with R bar. And this R bar, the position of this point with this element is making angle theta at a distance R. So the position represented with position vector, and the orientation of this point represented with theta, and the current direction element multiplied IDL. I write here this as current element. I write here it as IDL bar. So DL in the direction of current. So the magnetic field at this point due to small element is dB bar. This dB bar depend on the current in the wire, current in the conductor, and size of the element, and the distance of the point under consideration, and the point which is making the direction, the orientation of the point with respect to the small element. These are the factors which influence the magnetic field at this point. So now, the magnetic field at this point due to a current carrying conductor can be written as dB is proportional to IDL. I write here simply DL. So dB is proportional to DL, the first one, and also dB is proportional to the current, or sometimes you can write on IDL, and dB is proportional to or inversely proportional to square of the distance between the point and the element, and dB is proportional to sin theta. So you can just state that the magnetic induction, magnetic field due to a current carrying conductor at any point P is directly proportional to the length of the element, directly proportional to the current in the conductor, and inversely proportional to square of the distance between the element and the point, and it is also proportional to sine angle between the position vector and the small element. So if you combine all these, we can write down dB is proportional to I dl sin theta by r square. So this is only proportional. Suppose you want to make it equal, then we have to write down dB is equal to mu naught by 4 pi. This is some constant that depend upon the medium. For free space, I write here mu naught by 4 pi IDL sin theta by r square. So this is what is called the Biot-Savart law. So dB, what is the direction of this dB? Suppose if you observe the right hand rule, thumb rule, and see my thumb represents the direction of the current, and my fingers they're rotating in this direction, into the board, here it is, into the board, and here it is coming out. So like this. So here I just represent the direction in this, and here it comes out. So the field enters into this direction like this. This is how we can represent the direction of the field. Say, for example, I just represent here, this is a rectangular box you imagine. So if this represents the current IDL, and this represents R bar, and then what happened, and this is the X, and this is the point where it is under consideration, and this is making angle theta here, ideal, and this is the position vector here, point P. Then the direction of the field will be, this will be the magnetic field direction, dB. So this will be the direction, this direction. Just imagine. So dB is equal to mu naught by 4 pi ideal sin theta by r square. The direction will be this way. So we can also represent in a vector form. dB bar is equal to mu naught by 4 pi I into dl cross I into dl cross r whole by r cube. So this is in a vector form. We can represent this. Then what is the value of this? This mu naught is the permeability of the free space. So mu naught is equal to 4 pi into 10 power of minus 7 Henry per meter. So mu naught by 4 pi become how much? Mu naught by 4 pi become 10 power of minus 7. The Henry per meter. Then what is the unit of this mu naught? Mu naught we can write down. Unit of mu naught. Unit of mu naught. What is the unit of mu naught now here? dB, Tesla, r cube, or you can write down here dB, Tesla, meter square by ampere meter. Theta has no unit. So this is the unit of mu naught. So you can cancel this. So the unit of mu naught is equal to Tesla meter per ampere. This is also unit of mu naught. So now you can also write down this as Tesla meter per ampere. So this is how we can determine the value of the magnetic field. Now let us state the Biot-Savart law. Now let us observe the force on current carrying conductor placed in uniform magnetic field. Suppose you just consider that this is the uniform magnetic field B, and if the current carrying conductor I, length of the wire is L, which is making angle theta with the magnetic field, and then the force experienced by this conductor F is equal to I into dl cross, or simply you can write down L cross B. L cross B. This is the force experienced, and this force can be explained with the help of Fleming's left hand rule. See, just you imagine, you just keep your left hand, thumb, forefinger, index finger and middle finger, center finger. If C stands for the current, in the direction of current, and this stands for the direction of magnetic field B, and now the force acts in this direction along the direction of thumb. You see, central finger represents the direction of current, and this index finger, forefinger, indicates the direction of the magnetic field. Then what happens? Just you observe the fingers. This is the current and this is force, and this thumb represent the force in this direction. So the conductor moves in this direction. So the direction of this can be explained with the Fleming's left hand rule is equal to B I into LB sine theta. The direction can be explained in using Fleming's left hand rule. This is how we can represent. Suppose if the magnetic field is changed, if the current direction is changed like this or like this. So what I changed here? What I changed here? I just changed the direction of current in this way, in this direction, this way, and magnetic field direction didn't change, but the thumb pointing towards the board. The thumb pointing towards the board. So force act in the towards the board. So this is how the force on the current carrying conductor can be identified using Fleming's left hand rule. So Fleming's left hand rule. This is used in motor. And so if theta is 0, what happens? Force becomes 0. That is minimum. If theta is 90 degrees, angle between L and B, then force becomes maximum. That is equal to B I L. That means if the current carrying conductor carrying current length of the wire is normal to the direction of the field, then force is maximum. That is equal to B I L. And if the direction of the current and length of the wire parallel to the direction of the magnetic field, then force on the conductor is 0, that is minimum. If the conductor is making some angle theta, then the force will be ILB sine theta. So this is the cross product of the length of the current carrying conductor in the direction of current and magnetic field. This is how we can determine the force on current carrying conductor.
Welcome to online classes. I am Dr. M. Hemambara Reddy, JL in Botany, AP Residential Junior College, Nagarjuna Sagar. Today I am going to explain second year Botany, 5th unit, Molecular Biology. In this Molecular Biology lesson, Molecular Basis of Inheritance. So in this lesson, nucleic acids, that means DNA and RNA, structure, functions, I will explain. So in living organisms, DNA and RNA, these two together called as nucleic acids. DNA and RNA are the building blocks of genetic material. Genetic material means it is responsible for the transmission of characters from parents to offspring. That means from one generation to next generation. DNA is the genetic material in most of the organisms. That means generally except plant viruses, remaining organisms, DNA is the genetic material. RNA is the genetic material in some viruses. Generally, in plant viruses, RNA is the genetic material. RNA mostly functions as messengers. Generally, living organisms, we can observe three types of RNAs: mRNA, tRNA, rRNA. Later I will explain functions of RNAs. So, RNA also found in all organisms except some viruses. RNA in majority of the organisms functions as messengers. That means generally involved in protein synthesis. So DNA and RNA together are called a nucleic acid. These are the building blocks of genetic material. DNA is the genetic material in most of the organisms. RNA, that means ribonucleic acid, is the genetic material in some viruses. DNA means deoxyribonucleic acid. RNA mostly functions as messengers. Structure of polynucleotide chain. Polynucleotides are the polymers of nucleotides. Poly means many. DNA and RNAs are polynucleotides. That means DNA and RNA made up of nucleotides. So in DNA and RNA, so many number of nucleotides are there. That's why this DNA and RNA called as a polynucleotide chain. So nucleotide possess three types of components. That means a nitrogen base. This is nitrogen base. This is nitrogen base, sugar and phosphate. These three together are called as nucleotide. So this is one nucleotide, this is one nucleotide, this is one nucleotide, this is one nucleotide. So that so many number of nucleotides are there in DNA and RNA. That's why DNA and RNA are called as polynucleotide chain. You see here, phosphate is a connected fifth carbon of the deoxyribose sugar. That's why this is called as a 5 prime. So 5 prime is the starting point of the DNA chain. You see here, hydroxyl group is a connected third carbon of the sugar. This is treated as a 3 prime. So in DNA and RNA, each strand starting point is called as a 5 prime, ending point is called as 3 prime. This is very important. So DNA and RNA made up of nucleotides. That's why these are called as polynucleotide chains. Polynucleotides are the polymer of nucleotides. DNA and RNA are polynucleotides. So, you see, in nucleotide, three components are there: nitrogen base, pentose sugar. Sugars are different types based on number of carbons. Pentose sugar means the sugar possesses 5 carbons, 5 carbon sugar. For example, glucose, fructose, these are the hexose sugar, 6 carbon sugar. Whereas in DNA and RNA, we can observe pentose sugars. That means the sugars possess 5 carbons. So a phosphate group. So nitrogen base, sugar, phosphate, these 3 together called as nucleotide. So these 3, how together, you see here, this is sugar. That means 5 carbon sugar. Okay. So this is a nitrogen base. Nitrogen base is connected to that is a 5 carbon sugar. And you see this is a phosphate. So phosphate is connected to the sugar. So nitrogen base, this is sugar, phosphate. These 3 together called as nucleotide. So many number of nucleotides are there in DNA and RNA. That's why DNA and RNA called as a polynucleotide chain. This is structure of polynucleotide chain. Nitrogen base in nucleic acids, that means DNA and RNA. 2 types of nitrogen bases are there: purines, pyrimidines. Purines are called double ringed nitrogen bases. You see, this is one ring, this is one ring. This is one ring, this is one ring. So, double ringed nitrogen bases are called as purines. Single ringed nitrogen bases are called as pyrimidines. So, in nucleic acids, purines are 2 types: adenine, guanine. Whereas pyrimidines are 3 types: cytosine, thymine, uracil. So, in nucleic acids, total types of nitrogen bases are 5: adenine, guanine, cytosine, thymine, and uracil. DNA possesses 4 types of nitrogen bases. RNA also possesses 4 types of nitrogen bases. So in DNA, adenine, guanine, cytosine, thymine, these 4 nitrogen bases are present. Uracil is absent in DNA. In RNA, also possesses 4 types of nitrogen bases: adenine, guanine, cytosine, and uracil. Thymine is absent in RNA. That means thymine present in DNA, absent in RNA. Uracil present in RNA, absent in DNA. So adenine, guanine, cytosine, these 3 nitrogen bases are common both in DNA and RNA. Thymine present only in DNA, uracil present only in RNA. So thymine, that means called as a 5-methyl uracil, present only in DNA, and uracil only in RNA. So adenine, guanine, 2 belongs to purines, that means double ringed nitrogen bases. Cytosine, thymine, uracil, these 3 together called pyrimidines, that means single ringed nitrogen bases. So total 5 types of nitrogen bases are present in nucleic acids, that means DNA and RNA. Nitrogen base is linked to pentose sugar through N-glycosidic linkage to form nucleoside. So, nucleoside means nitrogen base and sugar, the two together called as nucleoside. That means here no phosphate. So nucleoside plus phosphate together called as nucleotide. So nitrogen base and pentose sugar is connected with one bond. That means N-glycosidic bond. Very important for MSET and NEET point of view. So the bond between sugar and nitrogen base, both the DNA and RNA, called as N-glycosidic bond. So this is pentose sugar, this is nitrogen base. These two connected with N-glycosidic bond. Structure of polynucleotide chain, nucleosides in RNA, nucleosides in DNA. Nucleosides in RNA, adenosine, guanosine, cytidine, uridine. These are the 4 types of nucleosides in RNA. In DNA, deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. These are the 4 types of nucleosides in DNA. Anyhow, nucleoside means nitrogen base and sugar, that too. You see, this is sugar, this is nitrogen base, adenine. This is guanine. This is cytosine. This is uracil or thymine. That means uracil present in RNA, thymine present in DNA. So, in RNA, 4 types of nucleosides, adenosine, guanosine, cytidine, uridine. In DNA, deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. So, these are the different types of nucleosides in DNA and RNA. Structure of polynucleotide chain. So, in polynucleotide chain structure, when we observe nitrogen base plus sugar plus phosphate group, 3 together, and it forms as a nucleotide. You see, this is phosphate. This is sugar, pentose sugar. That means 5 carbons are there. So, 1, 2, 3, 4, 5 carbons are there. This is nitrogen base. These 3 together, nitrogen base, sugar, phosphate, 3 together called as nucleotide. Whereas nucleoside possesses only sugar and nitrogen base only. Structure of polynucleotide chain. In RNA, each nucleotide has an additional hydroxyl group at second position of ribose sugar. 2 nucleotides are linked through 3' 5' phosphodiester bond to form dinucleotide. When more nucleotides are linked, it forms polynucleotide. So, in DNA and RNA, so how these nucleotides are connected, you see by this diagram. See, this is ribose sugar. Ribose sugar present only in RNA, whereas in DNA, deoxyribose sugar is there. So, In RNA, each nucleotide has an additional hydroxyl group at second position of ribose. Two nucleotides are linked through 3' 5' phosphodiester bond to form dinucleotide. When more nucleotides are linked, it forms polynucleotide. So this is dinucleotide. That means this is one nucleotide, this is one nucleotide. These two nucleotides are connected with this phosphodiester bond. So if more nucleotides are there, that is called as polynucleotide. So this is a 5', this is 3'. 5' is starting point, 3' is ending point of each chain of DNA or RNA. When we observe the innovations of DNA, Frederick Mischer in the year 1869 identified DNA and named it as nuclein. James Watson and Francis Crick proposed double helix model of DNA. It was based on X-ray diffraction data produced by Wilkins and Franklin. That means Messrs. Wilkins and Rosalind Franklin. So, when we observe the innovations, Frederick Mischer identified and named as a nuclein. Watson and Crick proposed double helix model of DNA. That means two strands of DNA arranged anti-parallel and twisted. That structure is called as a double helix model. So in DNA, two strands are there. The two strands arranged anti-parallel and twisted. So that is called as a double helix model. And it was based on X-ray diffraction data produced by Wilkins and Franklin. DNA is made up of two polynucleotide chains coiled in a right-handed fashion. Pitch of the helix, the length is 3.4 nanometers or 34 angstroms. Number of base pairs in each turn is 10. Distance between adjacent base pairs is 0.34 nanometers or 3.4 angstroms. You see, this is a DNA. This is one strand, this is one strand. Two strands are connected with hydrogen bonds. So DNA is made up of two polynucleotide chains. This is one chain, this is one chain. It is a right-handed fashion. So that means the right side twisted one. So this type of DNA is called as a B-form DNA. Pitch of the helix is 3.4 nanometers or 34 angstroms. That means one complete turn is called as pitch. One complete turn means here to here DNA shows a 360 degrees curve. Okay? Or from here to here, it is one complete turn. One complete turn means 360 degrees. Length of one complete turn is a 3.4 nanometers or 34 angstroms. That means a 10 angstroms is equal to 1 nanometer. So from here to here, 10 pairs of nucleotides are there. 10 pairs means 20. So, 10 pairs of base pairs. 20 base pairs means 20 nucleotides are there. Distance between adjacent base pairs. You see, this is one nitrogen base, this is one nitrogen base. The distance between two nitrogen base of the two same strands is 0.34 nanometers, equal to 3.4 angstrom. Here to here, one complete turn length is 34 angstroms. The distance between two successive nucleotides is 3.4 angstroms or 0.34 nanometers. Here to here, one complete turn, the angle is 360 degrees. The angle between two successive nucleotides is 36 degrees. Here to here, one complete turn, 360 degrees. The angle between two successive nucleotides is 36 degrees. So, DNA is made up of two polynucleotide chains coiled in a right-handed fashion. Pitch of the helix is 3.4 nanometers, equal to 34 angstroms. Number of base pairs in each turn is 10. Base pairs I am saying. Distance between adjacent base pairs is 0.34 nanometers or equal to 3.4 angstroms. When we observe structure of DNA, backbone of DNA is formed of sugar and phosphate. The bases project inside. The two chains have anti-parallel polarity. That is, one chain has the polarity 5' to 3' and other has 3' to 5'. You see here, this is the DNA. This is one strand, this is one strand. That means this is one strand, this is one strand. In these two strands, in this strand, this is 5' starting point. Opposite to here, this is 3', this is ending point. So, this is one strand and this is one strand. Two strands are connected with the anti-parallel. So, this is starting point, this is ending point. Here, this is starting point, this is ending point. So, two strands arranged anti-parallel. And these two strands connected with hydrogen bonds. That means, you see, this is nitrogen base, this is nitrogen base. Adenine pairs with thymine. Guanine pairs with cytosine. That means, adenine is complementary with thymine and vice versa. Guanine is complementary with cytosine. In between adenine and thymine, two hydrogen bonds are there. In between guanine and cytosine and vice versa, three hydrogen bonds are there. So, two strands of DNA connected with hydrogen bonds, that hydrogen bonds described by the scientist Fleming. So, backbone of DNA is formed of sugar and phosphate. The bases project inside. The two chains have anti-parallel polarity. That is, one chain has the polarity 5' to 3' and the other has 3' to 5'. The structure of DNA, the bases in two strands are paired through hydrogen bonds, forming base pairs. In between adenine and thymine, 2 hydrogen bonds. In between cytosine and guanine, 3 hydrogen bonds. That means purine comes opposite to a pyrimidine. This generates uniform distance between the two strands. So, in DNA, adenine is a complementary with thymine and vice versa. Cytosine is a complementary with guanine and vice versa. In between adenine and thymine, 2 hydrogen bonds. In between cytosine and guanine, 3 hydrogen bonds are there. So, you see, this is adenine and thymine. In between adenine and thymine, 2 hydrogen bonds. Whereas in between cytosine and guanine, 3 hydrogen bonds are there. So, this is one strand, this is one strand, 2 strands are connected with this hydrogen bonds. So, number of hydrogen bonds in between adenine and thymine and vice versa, 2 hydrogen bonds. In between cytosine and guanine and vice versa, 3 hydrogen bonds are there. So, adenine is a purine, thymine is a pyrimidine. That means a purine pairs with pyrimidine. Cytosine is a pyrimidine, guanine is a purine. So, pyrimidine pairs with purine. See the structure of DNA. So, 2 strands. The 2 strands of DNA is not a straight one. It is a twisted one. That's why DNA structure is called as a double helical structure. It is a right side twisted structure. When we observe these 2 strands, this is one strand and this is one strand. In this strand, you see, this is phosphate group. This is phosphate group, phosphate group, phosphate group. This is sugar, phosphate sugar, phosphate sugar, phosphate. That is pentose sugar. Pentose sugar, pentose sugar. That means a 5-carbon sugar. Pentose means 5-carbon sugar. These are the nitrogen bases. So, nitrogen base, pentose sugar and phosphate. These 3 together are called as a nucleotide. So, phosphate, pentose sugar, nitrogen base. So, this is one nucleotide, this is one nucleotide, this is one nucleotide. So many number of nucleotides are there. That's why this DNA is called as polynucleotide chain. So, between 2 strands, hydrogen bonds are there in between nitrogen bases. So, between adenine and thymine, 2 hydrogen bonds. Between guanine and cytosine, 3 hydrogen bonds are there. Erwin Chargaff proposed one rule in DNA. In DNA, the proportion of adenine is equal to thymine and proportion of guanine is equal to cytosine. That means always adenine complementary with thymine. Guanine is complementary with the cytosine. So, number of adenines is equal to number of thymines. Number of guanines is equal to number of cytosines. So, adenine plus guanine is equal to thymine plus cytosine, or adenine plus guanine by thymine and cytosine is 1. That means number of purines is equal to number of pyrimidines. That is called as Chargaff principle or Chargaff rule. So, adenine is complementary with thymine. Guanine is complementary with cytosine. So that adenine plus guanine equal to thymine plus cytosine. So this is proposed by Chargaff. That's why this rule is called as Chargaff principle. That means the number of purines are equal to number of pyrimidines. Regarding this DNA, lambda 174, it is a bacteriophage. Bacteriophage means it is a virus. Virus attacks the bacteria is called as bacteriophage. Has 5,386 nucleotides are there. That means the number of nucleotides are fixed in lambda 174 bacteriophage. Each bacteriophage possesses 5,386 nucleotides. Where the bacteriophage lambda has 48,502 base pairs. So this is bacteriophage lambda has 48,502 base pairs. So in this bacteriophage, DNA is a single strand. Here, DNA is a double strand, so that 48,502 base pairs are there. In Escherichia coli has 4.6 into 10 to the power of 6 base pairs. E. coli means Escherichia coli, generally found in human beings' intestine. So each E. coli possesses 4.6 into 10 to the power of 6 base pairs. That means number of nucleotides is different in different organisms. Haploid content of human DNA. That means in human beings, each cell possesses 3.3 into 10 to the power of 9 base pairs. Haploid content of human DNA. That means one set of chromosomes. So each cell in human being possesses 2 sets are there. Okay. So haploid content of human DNA is 3.3 into 10 to the power of 9 base pairs. So in human beings, all the cells are diploid. That means that 2 sets of chromosomes are there. 2 sets possess 46 chromosomes. That's why in human being, the number of chromosomes is 2n equal to 46. 2n means 2 sets. Each set possesses 23 chromosomes. In these 23 chromosomes, 3.3 into 10 to the power of 9 base pairs are there. So lambda 174, 5,386. Bacteriophage lambda has 48,502 base pairs. Escherichia coli possesses 4.6 into 10 to the power of 6 base pairs. Haploid content of human DNA is 3.3 into 10 to the power of 9 base pairs. Calculation of length of DNA. So length of DNA is number of base pairs into distance between 2 adjacent base pairs. Example, number of base pairs in human beings 6.6 into 10 to the power of 9. Hence, the length of DNA is 6.6 into 10 to the power of 9 into 0.34 into 10 to the power of 9. So, 2.2 meters. That means in each cell of human being, the DNA length approximately 2.2 meters. So the cell is microscopic. We can't observe a single cell with our naked eye. But in that single cell, 2.2 meter length of DNA is there. That means the DNA is highly twisted structure. That twisted DNA is located in a cell. In E. coli, length of DNA is 1.36 millimeter. That means 1.36 into 10 to the power of 3 meters. So the number of base pairs is 1.36 into 10 to the power of minus 3 by 0.34 into 10 to the power of minus 9. So equal to 4 into 10 to the power of 6 base pairs. So DNA, the length we can calculate that by number of base pairs into distance between two adjacent base pairs. So the distance between two adjacent base pairs is 0.34 nanometers or 3.4 angstroms. So packing of DNA helix. How DNA is packed in a cell. That means in human beings, already I was explained, the cell is microscopic, we can't observe with our naked eye. But each cell possesses a 2.2 meter length of DNA is there. The DNA is formed as a twisted structure in each cell. Packing of DNA helix. In prokaryotes means in some of the organisms, a conspicuous nucleus is absent. That means there is no nuclear envelope. Such a cells are called as prokaryotic cells. These prokaryotic cells are phylogenetically primitive. Whereas phylogenetically evolved cells are called as eukaryotes. Eukaryotes, the cells possess a conspicuous nucleus is there. That means nuclear envelope is there. So example for prokaryotes, bacteria and blue-green algae. These are the belongs to prokaryotes. So in prokaryotes, example Escherichia coli, the DNA is not scattered throughout the cell. DNA is negatively charged. So it is held with some positively charged proteins to form nuclei. So DNA connected with a positively charged proteins so that it forms as a nuclei. So this is bacteria cell. You see this is a nuclei. So DNA is associated with some proteins. So prokaryotic cell, this is a plasmid. Plasmid means this is a circular DNA. Whereas chromosomal DNA, it is not a circular DNA. It is a double strand helical one. It is called as chromosomal DNA or called as nuclei. Whereas plasmid is a circular DNA. Plasmid also double strand structure. So in prokaryotes, the DNA is not scattered throughout the cell. DNA is negatively charged. So it is held with some positively charged proteins to form nuclei. In eukaryotes, there is a set of positively charged basic proteins called histones. Histones are rich in positively charged basic amino acid residues, lysine, arginine. 8 histones form histone octamer. Negatively charged DNA is wrapped around histone octamer to give nucleosome. You see, this is a nucleosome structure. In this nucleosome, histone core is there. This is histone core. In this histone core, 8 histones are there. So, H2A, H2B, H3, H4. 4 types of histones are there. H2A, H2B, H3, H4. So, in the histone core, 2 H2As, 2 H2Bs, 2 H3s, 2 H4s. So, total 8 histones are there. 4 types are there. Each type 2 histones are formed. This is called as histone core. Histone core is wrapped by DNA. Histone core wrapped 2 turns with the DNA. So, in eukaryote, there is a set of positively charged basic proteins called histones. Histones are rich in positively charged basic amino acid residues, lysine and arginine. The question came several times in MSET and NEET also. So, the histones charged basic amino acid residues, lysine and arginine. 8 histones form histone octamers. Negatively charged DNA is wrapped around histone octamer to give nucleosome. So, this is nucleosome. So, this is histone core. This is a DNA wrapped 2 turns. This is 1 turn and this is 1 turn. So, here 1, 2, 3, 4, 5, 6, 7, 8. 8 histones are there. That's why it is called as histone octamer. 4 types are there. H2A, H2B, H3, H4. So, each 2 types are there. 2 H2As, 2 H2Bs, 2 H3s, 2 H4s are there. A typical nucleosome contains 200 base pairs are there. So, this is one nucleosome. This nucleosome possesses 200 base pairs. Therefore, total number of nucleosomes in human, you see, E cell possesses 6.6 into 10 to the power of 9 base pairs. So, each nucleosome possesses 200. So that 3.3 into 10 to the power of 7 nucleosomes are present in each cell of human being. So, this is one nucleosome, this is one nucleosome. 2 nucleosomes are connected DNA. The DNA is called as a linker DNA or also called as spacer DNA. So, this is one nucleosome, this is one nucleosome. Each nucleosome wrapped with 2 terms of DNA. Between these 2 nucleosomes, DNA is there. This DNA is called as a linker DNA or also called as spacer DNA. So, a typical nucleosome contains 200 base pairs, so that in human being, each cell possesses 3.3 into 10 to the power of 7 nucleosomes are there. Nucleosomes constitute the repeating unit to form chromatin. Chromatin is the thread-like stained bodies. Nucleosome in chromatin is beads on string. So, this is one nucleosome, this is nucleosome, this is nucleosome, this is nucleosome, this is nucleosome. So, nucleosomes constitute the repeating unit to form chromatin. So, it is a twisted structure. chromatin is the thread-like stained bodies. Nucleosome in chromatin is a beads on string. So, chromatin is packed so that it forms chromatin fibers. Chromatin fiber twisted and it forms as a coiled and a condensed at a metaphase stage. And this coiled condensed structure compactly together and it forms as a chromosomes. So, in a normal cell at a metaphase stage, that's why metaphase stage is the most suitable stage to observe chromosomes. So, chromatin packed and it forms as a chromatin fiber, and chromatin fibers coiled and condensed and it forms as a chromosome. So, higher level packaging of chromatin requires non-histone chromosomal proteins. So, to form chromatin requires non-histone chromosomal proteins. So, in DNA, based on staining, we can observe two different parts, euchromatin, heterochromatin. So, euchromatin means loosely packed, transcriptionally active region of chromatin. It stains light. That is called as euchromatin. You see, this is euchromatin. Heterochromatin means densely packed, inactive region of chromatin. It stains dark. So generally, chromosomes are stained with acetocarmine pigment. When stained with acetocarmine, lightly stained one is called as euchromatin, thickly stained one is called as heterochromatin. Euchromatin is a loosely packed, transcriptionally active region of chromatin. It stains light. Heterochromatin, densely packed, inactive region of chromatin. It stains dark. Very important for 2 marks. What is meant by euchromatin and what is meant by heterochromatin. So, euchromatin is genetically active. Heterochromatin is genetically inactive or passive. So that in chromosome we can observe euchromatin and heterochromatin regions. So up to here I was explaining structure of DNA. Now the following animation you see so that you can understand structure of DNA in detail. Thank you.
Topic of this presentation is structure of DNA. First of all, we must learn basic concepts related to this topic. So we will start this presentation with nucleotides, as these are units or building blocks of nucleic acids, RNA and DNA. A nucleotide consists of 3 components: nitrogenous base, phosphate group and pentose sugar. As shown in the flowchart here, there are 2 types of pentose sugars, ribose and deoxyribose. Ribose sugar is present in RNA, whereas deoxyribose sugar is present in DNA. As shown in the diagram, a pentose sugar is a 5-carbon ring, and these carbons are named as 1' carbon, 2' carbon, likewise 5' carbon. If you know this structure is clear to you, then you will be able to understand when we say 3' position or 5' position of DNA strand in the discussion ahead. One more important point, compare the structure of ribose sugar and deoxyribose sugar. So we find hydroxyl group at 2' position in ribose sugar whereas hydrogen group at 2' position in deoxyribose sugar. Now this presence of hydroxyl group at 2' position in ribose sugar makes it more reactive than deoxyribose sugar. Therefore, RNA is more reactive than DNA, as we already know. Now the nitrogenous bases. So two types of nitrogenous bases, pyrimidines and purines, exist. And here we have shown the structure skeleton of these nitrogenous bases. Purine is the heterocyclic ring of 9 atoms of carbon and nitrogen, whereas skeleton of pyrimidine is a heterocyclic ring of 6 carbon and nitrogen. Some modifications occur in these rings to form various types of bases as shown here. 2 types of purines, adenine and guanine, and 3 types of pyrimidines, thymine, cytosine and uracil, are seen in these nucleic acids. With the help of these diagrams, we will understand difference between nucleoside and nucleotide. See this diagram of nucleoside. Here a nitrogenous base is attached at 1' position of a pentose sugar with the help of N-glycosidic bond. So when nitrogenous base is attached to pentose sugar, then this combination is called a nucleoside. And when a phosphate group joins this nucleoside, then a nucleotide is formed. Now see this structure. So here this is the pentose sugar, and at 1' position a nitrogenous base is attached through N-glycosidic bond, and at 5' position a phosphate group is attached through ester bond. So this is the structure of nucleotide, and combination of pentose sugar and nitrogenous base is the nucleoside. So this is the difference between these two units. We will now understand formation of phosphodiester bond between two nucleotides with the help of this diagram. Now see the alignment of first nucleotide. This is the 5' position of first nucleotide with phosphate group, and this is the 3' position with hydroxyl group. And the second nucleotide is also aligned in the similar way. This is the 5' position with phosphate group, and this is the 3' position with hydroxyl group. Now hydroxyl group of 3' position of nucleotide first will react with OH group of phosphate at 5' position of nucleotide second. As a result, a molecule of water will be eliminated and phosphodiester bond will be formed between 3' position of nucleotide first and 5' position of nucleotide second. So in this way we have shown you the formation of dinucleotide chain, and this is the 5' position of dinucleotide chain, and this is the 3' position of the dinucleotide chain. So we can say that this dinucleotide chain is running from 5' to 3' direction. So if our basics are clear like this, then it will help you understand and retain the structure of DNA very well. With the help of this diagram, we will now understand the structure of a polynucleotide chain in detail. So here we have shown this polynucleotide chain, and this is its 5' end with phosphate group, and this is the 3' end of this polynucleotide chain. These are the phosphodiester bonds between the nucleotides in this polynucleotide chain, and a nitrogenous base is attached to each nucleotide, and these nitrogenous bases are protruding into the right side of this polynucleotide chain. And in this polynucleotide chain, this is the 5' end and this is the 3' end. So we can say that this polynucleotide chain is running from 5' to 3' direction. So after understanding this structure of polynucleotide, it will be very easy to understand the concept of anti-parallel orientation of DNA strands in a DNA duplex. One more thing we want to tell to our students that in this polynucleotide chain, this pentose sugar and phosphate group, these two together make the backbone of this polynucleotide chain, and nitrogenous bases remain free to form hydrogen bonds with the nitrogenous bases of the other strand of DNA to make the double-stranded structure in a DNA duplex. We will now understand making of double-stranded DNA and its further coiling. Now, two strands align themselves in such a way their bases lie opposite to each other, and strands also follow anti-parallel orientation. Here, meaning of being anti-parallel is that if one strand is running from 5' to 3' direction, then the other strand runs from 3' to 5' direction. And this bases also follow complementarity rule, which means that adenine always pairs up with thymine with the help of 2 hydrogen bond, and cytosine pairs up with guanine with the help of 3 hydrogen bonds. In this way, following these rules, a duplex or double-stranded DNA is made. Now we will compare this double-stranded DNA with a rope ladder. Visualize that you are holding a small rope ladder in your hands and twisting it clockwise or anti-clockwise. Then if ladder remains straight during twisting, then it will result in symmetrical coil with equal sized grooves in its sides. But if the rope ladder gets tilted a bit during twisting, then it will result in an asymmetrical coiling with grooves of unequal size in its sides. Likewise, DNA duplex gets tilted in the same way during coiling, due to which we observe major and minor grooves in its structure. Finally, we will now understand the structure of DNA with the help of this diagram. Here, major and minor grooves are shown in the diagram, and we have already told how these grooves are formed during coiling. And see this anti-parallel orientation of strands. One strand is running from 5' to 3', while the other strand is running from 3' to 5' direction. Thickness of this duplex is 2 nanometer. And here one complete turn of DNA strand is shown, and with one major and one minor groove. Length of one complete turn is 3.4 nanometer, and there are 10 base pairs in one turn. 10 base pairs are not shown in this diagram, so please don't count the base pairs in this one turn. Now by calculation we find that there is a distance of 0.34 nanometer between two base pairs. So this is the structure of DNA, and we hope you have understood it very well. Please check your understanding by answering these questions.